So yes, very good afternoon to one and all. Yesterday we have dealt with shortened type of questions from the chapter Principles of Inheritance and Variation, wherein we have dealt clearly with the concepts of law of dominance from monohybrid cross, followed by test cross, and then we have dealt with Mendel's advantages of choosing Pisum sativum as model organism by Mendel, and then we have seen law of segregation and law of independent assortment, comparative analysis. We have also looked into the differences between dominant allele, recessive allele, homozygous, heterozygous, monohybrid cross and dihybrid cross. And then we have looked upon codominance, chromosomal mutations and gene mutations. Now we are going to begin with molecular basis of inheritance. Very important questions asked multiple times. It is about nucleosome. Nucleosome model is also important for your MSET and NEET. Okay? So learn the concept in such a way so that you will be prepared for both subjective and objective type of answering. Here we go with nucleosome model. First look at the importance of the question. I will explain you in brief and then we will go through. So first, if you see, this question has appeared in multiple years like June 2024, 2023, 2022 and May 2022 as well. So here the negatively charged DNA is wrapped around positively charged histone proteins. As such, the chromatin material will get condensed to chromosome. Okay? So the negatively charged DNA in the form of chromatin is wrapped around positively charged histone proteins. So in a single octamer we have two pairs of 4 histone proteins. Those are H2A, H2B, H3 and H4. As such, the DNA is wrapped around the histone octamer. From one histone octamer to other histone octamer, the DNA which is connecting is addressed as linker DNA. This linker DNA is holded with H1 histone proteins, and this forms one nucleosome model. A single nucleosome will have 200 base pairs length of DNA. 200 base pairs length of DNA. Clear, right ma? So here when you see the negatively charged DNA wrapping around the histone octamer, this complete chain will be visible as beads on the string arrangement. Okay? So this is the concept of nucleosome model. Now we will look into the systematic presentation of the answer. Here you see ma, first nucleosome is a fundamental structural and functional units of chromatin that is found inside the nucleus. This is found inside the nucleus of eukaryotic cell. The histone proteins will organize, they are organized to form a core of 8 molecules. This forms a core of how many molecules? 8 molecules. These 8 molecules are known as what ma? Histone octamer. Okay. Histone proteins are positively charged amino acids. This histone protein is positively charged because it consists of positively charged amino acids which are lysine and arginine. Okay, lysine and arginine, these are positively charged amino acids. The histone octamer is made up of 2 pairs of 4 histone proteins which are H2A, H2B, H3 and H4. And then you see negatively charged DNA is wrapped around positively charged histone octamer. Okay, negatively charged DNA is wrapped around positively charged histone octamer, which gives out what beta? Nucleosome model. Each nucleosome will consist around 200 base pairs length of DNA helix, and then H1 histone protein will lie outside the core, and this will help to seal the DNA which turns by binding where DNA enters and exits the core histone octamer. The DNA strand between two nucleosomes is known as linker DNA. You have to highlight these terms when you are presenting the answer, because examiner will be looking for those terms. Okay. Here, next is under an electron microscope, chromatin appears as what? Beads on string arrangement or beads on string structure, where each bead is a nucleosome. These nucleosomes are repeating units that together form a chromatin structure within the nucleus. The beads on string structure further coils and it gets condensed to form what? Chromatin fibers. Finally, this folds into what? Chromosomes. Finally, this folds into chromosomes during cell division. Fine, right? This is all about nucleosome model. First, if you keep the diagram as your reference, then it will be easy for you to elaborate this diagram point wise. Okay. Now, next you see ma, what is the difference between DNA and RNA? Whenever they are asking you the differences or comparative analysis, it is better to provide a tabulated form, okay, like this, a table. Fine. DNA and RNA. DNA, you see, this consists of two strands of nucleotides which are forming a double helix. RNA, this consists of single strand of nucleotides. It has single strand of nucleotides, and then this is found in the nucleus. They are found mainly inside the nucleus with small amounts of mitochondria and chloroplast as well. DNA is present in chloroplast and mitochondria as well along with the nucleus. RNA is found in the cytoplasm and small amounts of it is found in the nucleus as well. Contains deoxyribonucleoside sugar or deoxyribose sugar, and this has ribose sugar. Both are pentose sugars only, but in DNA we have deoxyribose sugar, in RNA we have ribose sugar. And then you see here ma, pyrimidines. Pyrimidines we have thymine, cytosine. Okay, pyrimidines we have what? Thymine and cytosine. Thymine and cytosine. Here we have pyrimidines like uracil and cytosine. Base pairing, if you see, base pairing is between adenine thymine and guanine cytosine. Here it is binded with 2 hydrogen bonds. Guanine cytosine is binded with 3 hydrogen bonds. In RNA, if you see, base pairing is in between adenine and uracil with 2 hydrogen bonds, guanine and cytosine with 3 hydrogen bonds. Next, this serves as genetic material in almost all the organisms, whereas RNA, it is usually, we have two types of RNA, right? One is non-genetic RNA and the other is genetic RNA. Usually it is non-genetic but acts as genetic material only in some viruses. And then here we go, this does not directly participate in protein synthesis. Okay, it will not directly participate in protein synthesis, but this is directly involved in protein synthesis. Okay, this is directly involved in protein synthesis as mRNA, tRNA and rRNA. This exists as one type only, DNA, whereas RNA exists in 3 types, where we have mRNA, tRNA and rRNA, where mRNA is synthesized by RNA polymerase II, tRNA is synthesized by RNA polymerase III, rRNA is synthesized by RNA polymerase I. Clear till here? Any doubt anywhere? Yeah. Next, what are the contributions of George Gamow, Hargobind Khorana and Marshall Nienberg in deciphering the genetic code? The question about genetic code is also important, but it is not that multiple times repeated question. So, here if we see the contributions in deciphering the genetic code, what are the contributions? First you see about George Gamow. George Gamow has proposed a genetic code which must be what? A triplet in nature. Okay, this is triplet in nature, which means that 3 nucleotides together code for 1 amino acid. He mathematically suggested a combination of 4 bases which are taken 3 at a time. So, here you will get 4 to the power of 3, overall how many codons beta? 64 codons, which will code for how many amino acids? 20 amino acids. It will code for all the 20 amino acids. Next, This idea laid the foundation for understanding the triplet nature of the genetic code. Okay, this laid the foundation to understand the triplet nature of genetic code. Next, Hargobind Khorana or H.G. Khorana. So, Hargobind Khorana, he has chemically synthesized RNA molecules with specific and known sequences of the bases. He used both homopolymers and heteropolymers. Best example he has taken is uracil. Homopolymers, for example, U continuous chain, you will have copolymers which have uracil along with cytosine or cytosine along with adenine. This will determine the code with corresponding to the amino acid. His experiments provided definite proof specific codon amino acid relationship. Okay, he has decoded that, like which codon is coding for what amino acid, confirming what specificity, universality of the genetic code. Next, it is the scientist Marshall Nienberg who developed cell-free synthesis system. Okay, this enabled to directly observe protein formation. This will directly observe what protein formation from synthetic RNA template. He discovered poly-U-RNA, polyuracil chain, poly-U-RNA directed the synthesis of phenylalanine, providing UUU codes. For phenylalanine, the first codon E is ever deciphered. Next, later using the enzyme name is polynucleotide phosphorylase. Using this enzyme, he and his colleagues synthesized RNA with defined sequences. This leads to complete decoding of genetic code. Fine, right? So, here you see you have to elaborate the genetic code, how the genetic code is deciphered by George Gamow, Hargobind Khorana and Marshall Nienberg. Each and every point is important, beta. If you learn these statements clearly, it will be beneficial for you to answer the multi-statement questions also, if at all it appears in EAMCET and NEET. Now, the question is about how many types of RNA polymerases exist in the cell. Write their names and functions. So, we have 3 types of RNA polymerases, yes or no? The only RNA polymerase that exists in a prokaryotic cell is RNA polymerase 2. But here we have in eukaryotes, we have 3 types of RNA polymerases inside a eukaryotic cell. And every RNA polymerase is responsible for transcribing a specific set of RNAs. First you see RNA polymerase 1. RNA polymerase 1 will transcribe rRNA genes. Okay, it will transcribe rRNA genes. The products synthesized are 28S, 18S and 5.8S rRNAs. Next, RNA polymerase 2. Okay, RNA polymerase 2. This will transcribe structural and protein coding genes. Okay, they will transcribe structural and protein coding genes. Products synthesized for this. This will produce what? Precursors of mRNA or hnRNA, which will later form mature mRNA. This will later form what, ma? Mature mRNA, and this synthesizes some small nuclear RNAs. Those are called as snRNAs. Next, RNA polymerase 3. What does RNA polymerase 3 do? This will transcribe small RNA genes. They will transcribe small RNA genes which will produce tRNA, 5S rRNA, snRNAs. Okay. So, these are the products that are synthesized from RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Clear till here? Any doubt anywhere, ma? Yeah. Next we see, define transformation in Griffith's experiment. Discuss how it helps in identification of DNA as genetic material. So, here if you see, ma, bacterial transformation first. Bacterial transformation is a process by which genetic material is transferred from one bacterial cell or one bacterial cell, that is donor, to the other bacterial cell, that is recipient cell, through the uptake of naked DNA fragments. Okay. This is through the uptake of naked DNA fragments, resulting in change in the recipient's genotype and phenotype. Griffith has conducted experiments on Streptococcus pneumoniae. Okay, this is the bacteria which causes pneumonia in mice. Okay, the bacterium will exist in two forms. One is smooth form, that is smooth strain, which is virulent form. See, here the two strains of bacteria is what ma? One is S strain. S strain has a smooth coat over the bacteria. This smooth and shiny coat will protect the bacteria from host immune system. Whereas R strain, if you see, R strain is R strain doesn't have any smooth coat over it. This doesn't have any smooth coat over it. Okay, as such, R strain is subjected to the host immune system easily. So here you see the bacterium exists in two forms, a smooth strain which is virulent, and this has what? A polysaccharide capsule. Okay, and rough strain or R strain which is non-virulent, this lacks what? Capsule. Fine, right? Here we go. The experiment goes like this. First, when the virulent strain is injected to the mice, that is S strain, mice will die. And then when non-virulent strain that is injected to the mice, mice lives. And when S strain bacteria is heat killed and that S strain is injected to the mice, mice lives. This heat killed S strain, when it is mixed with R strain, again mice dies. This last experiment confirms that there is some chemical substance that is being transferred from heat killed S strain to R strain, which is making the R strain virulent and killing the mice. Okay, so that is what is proved in the transforming principle by by Griffith from the model organism Streptococcus pneumonia. So when live S strain is injected to the mice, mice died, and this is how you have to elaborate the answer. In the last point of the answer, you have to prove that when a mixture of heat killed S strain and R strain were injected, the mice died and the living S strain was recovered by proving what? Transforming principle from the dead S strain, which is converted to R strain, into virulent form. Clear? Any doubts anywhere ma? And here we go with write the important features of the genetic code. What are the important features of genetic code? Which is observed in like Feb, March 2024 and May 2022, August 2022, March 2018 and 2015. These many years the question has appeared. So the question about genetic code has appeared many times. When you have to choose, if you are in hurry of your preparation and you are quickly choosing the questions, then you have to choose the specific features of the genetic code more than deciphering the genetic code. Okay, this question is very important. Which like important features of genetic code include, first is it is a triplet. Each amino acid is encoded by a sequence of triplet nitrogen base. Okay, it has triplet nitrogen base, that is 3 nitrogen base codon on the mRNA. Out of how many? Out of 64 codons, only 61 will code for amino acids and 3, for example, these are stop codons which have UAA, UAG and UGA. These are stop codons. Okay, and then you see unambiguous and specific. Each codon will code for only one specific amino acid. They will code for only one specific amino acid. This ensures there is no ambiguity in protein. Okay, there is no ambiguity in protein synthesis. Next, degenerate code. Some amino acids are specified by more than one codon. Okay, some amino acids are specified by more than one codon. For example, leucine and serine. And then non-overlapping and comma-less. This is one more feature. Okay, non-overlapping and comma-less, where codons are read continuously in mRNA. They are read continuously in mRNA without overlapping and without punctuation. Without overlapping and punctuation marks. Next, genetic code is nearly universal. The genetic code is almost same in all the organisms, be it a prokaryotic cell or a eukaryotic cell. So, from bacteria to humans, all the genetic code is what? Universal. It is specific for a single amino acid. For example, if you see U, this codes for what? Phenylalanine in both prokaryotes and eukaryotes. Minor exceptions are there, they will exist in mitochondrial and protozoan codons. Next, start codon. AUG acts as what? Initiator codon. AUG acts as initiator codon. The signaling in the beginning of translation, they will help signaling in the beginning of the translation. It is also codes for what? Amino acid methionine. Okay. This also codes for amino acid methionine. So, it is of what function? Dual function. Next, stop codons. We have 3 stop codons. Stop codons are also addressed as termination codons. As such, we have UAA, UAG and UGA. They will signal the end of the protein synthesis. Fine? Clear till here? Any doubt anywhere? So, what are the specific features of the genetic code? We have, it is a triplet code, unambiguous and specific, degenerate code, non-overlapping and commaless, nearly universal, and it is a start codon, stop codon. About start codons and stop codons you have to give at the end. So, this is the complete content that you have to deliver for what? Genetic code features. Last but not the least, about the schematic or diagrammatic representation of gene expression by using what model? Lacoperon model, which is very easy to understand, very easy to present. It will not consume much time of yours during the exam time, and gene expression or Lacoperon model is very much important for your NEET and MCET as well. So, exercise the concept in that way. If you look into the IPE, how many times the question is asked? Like it is asked in May 2022, May 2018, 2016 as such. So, here you see ma. Yeah. We have promoter followed by I gene, promoter and operator. We know that to the promoter region all the time RNA polymerase will come and bind, okay, and I gene will synthesize repressor. So, first come first to the promoter when RNA polymerase is binded, this will start or initiate the process of transcription, and I gene will be releasing out the repressor mRNA. This will give out the repressor. Repressor will go and bind to the operator region. Repressor will bind with what region ma? Operator region. Here again to the promoter region RNA polymerase will bind, and if repressor is present, lactose will act as repressor here, okay. So, if repressor is going and in the absence of inducer, what happens is, what is the inducer over here? Inducer is lactose, fine. In the absence of lactose, whenever the repressor is binding with the operator, what happens? This process will get completely inhibited, and after operator you have 3 structural genes we have discussed. Those are what? Z gene, Y gene and A gene. Z gene will code for beta galactosidase, Y gene codes for permease and A gene codes for trans-acetylase. Here we go with this. In the presence of inducer, so whenever you are presenting this question, that is gene expression, you have to give out 2 things. One is in the absence of inducer what is happening, in the presence of inducer what is happening. In the presence of inducer here is again lactose. In the presence of inducer, wherein I gene, of course it will synthesize repressor, right. This repressor will be binded to the inducer, that is lactose. Okay, repressor is binded to the inducer. As such, here operator region is not blocked, so the structural genes will exit their functions. they will exhibit their functions and they will give out polycistronic expressions, as here we have 3 different structural genes. Okay. So, we have promoter, and lastly if you can include terminator also you can include, but then in between you have to give out 3 different structural genes like Z, Y, A. What is it coding for exactly? And after translation this will give out polycistronic expression. Polycistronic expression. Fine, any doubt anywhere? So that's all for today's session beta. What are the questions that we have discussed today? Nucleosome model, without any exception you have to go through nucleosome model in the mol bio, and then characteristic differences between DNA and RNA you have to go through, after which this is optional, this is up to you, deciphering genetic codon, and types of RNA polymerases is very important.